Welcome back to The Explainer. Today, we're setting the stage for an absolutely fascinating thought experiment. We're going to look at a prompt, a string of concepts that honestly sounds chaotic on the surface, almost like a total word salad. But as we unpack the layers together, you're going to see just how deep this rabbit hole goes. We've got biology, we've got complex mathematics, and we've got cutting-edge artificial intelligence all mashed together into one wild concept. So what could possibly connect mouse-licking behavior to AI scaling laws and human just-in-time compilers? I mean, it literally sounds like the setup to a very niche, highly nerdy joke, doesn't it? But we're going to treat this premise like a detective case that we need to solve together. We have a handful of very specific clues, and our job is to figure out how they could conceivably fit into one grand, futuristic picture. All right, to keep us on track, here's our roadmap for today. Section 1. The bizarre science mystery. Section 2, decoding biology and math. Section 3, unpacking DeepMind AI models. And finally, Section 4, the hypothetical lab workflow. Okay, let's dive into Section 1, the bizarre science mystery, and officially kick off our deconstruction. So here's the deal. According to current scientific literature, there is no real-world research or framework that organically connects all of these exact elements together. We're essentially analyzing a highly complex test string or a very creative synthesis that perfectly blends distinct concepts from math, animal behavior, and computer science. But here's the catch. While the overarching framework connecting them might be a hypothetical thought experiment, the individual puzzle pieces we're about to examine are completely 100% real. Let's look at those real-world puzzle pieces right now. Moving right along to Section 2, decoding biology and math. Let's ground ourselves in these foundational elements first to see exactly what we're dealing with. First up, we have the Lambert W function. Yeah, it sounds a little intimidating, but it's a very real mathematical function, specifically used to solve equations where an unknown variable appears as both a base and in an exponent. Think of something like x times e to the power of x equals y. Now, you might be wondering, why does this matter? Well, in the real world, biologists and physicists use the Lambert W function to model physical realities. They use it to calculate growth rates, to map out enzyme kinetics, and most importantly for our mystery, to model thermodynamics. Keep that word in your back pocket. Thermodynamics. Next, we're going to completely switch gears from complex math to animal behavior, allo-grooming. This is the scientific term for mouse or rat licking and grooming behavior. In modern neuroscience and animal behavior labs, researchers closely track exactly when these animals groom each other. Why? Because it's an absolutely crucial way to study social bonding, dopamine release, and even empathy. And what's really wild is that real labs actually use Google DeepMind's computer vision tools to meticulously track and map these exact interactions between mice. So, how on earth do we combine the math of thermodynamics with mouse licking? Well, some seriously advanced neuroscientists use what are called maximum entropy or thermodynamic frameworks to predict animal behavior. They literally treat a group of mice in an enclosure like physical particles bouncing around in a physics system. By applying these thermodynamic equations, which remember can be solved using our Lambert W function, they try to predict exactly when certain animal actions like grooming will trigger. It's an incredibly effective analogy for visualizing how advanced data science tackles biological behavior. Now, what's really interesting here is how they visualize this data. Researchers use standard scatter plots. On the x-axis, you might plot the temperature or the energy state of our mouse system. And on the y-axis, you plot the time until the licking actually starts. By plotting these variables against each other, scientists can look for visual patterns or clusters. This basically proves that their thermodynamic models are hitting the nail on the head when predicting biological behavior. All right, Section 3, unpacking DeepMind AI models. We've got our biology and our math down, so it's time to transition into the highly technical artificial intelligence clues embedded in this mystery. One of the standout terms in our mystery string is alpha genome. Now, if you follow AI at all, you know this aligns perfectly with Google DeepMind's naming conventions for their biological AI models. We already have AlphaFold, which completely revolutionized our understanding of proteins, and AlphaMissense for genetics. Alpha genome generally refers to an AI framework specifically designed to map out and deeply understand complex genomic sequences. It is, essentially, the ultimate biological decoding tool. Next clue, the famous chinchilla scaling laws. In the real world, this was a landmark DeepMind paper proving that for peak AI performance, you absolutely must scale an AI model's size and its training data equally. But our mystery prompt throws in a crazy sci-fi twist, the chinchilla scaling laws of ICE, or intelligent coordinate expansion. So we're taking a real, proven rule for scaling language AI and hypothetically applying it to expanding spatial or biological data coordinates. Which brings us to section 4, the hypothetical lab workflow. We've gathered all our clues, now let's put it all together into one futuristic DeepMind lab workflow. If a lab were to actually build a project connecting all these disparate terms, it would beautifully flow through 4 steps. Step 1, the behavior phase, watching the mice. Step 2, genetic mapping, diving into their DNA. Step 3, scatterplot visualization, crunching the data. And Step 4, scaling in human jet, taking the findings to the ultimate level. Let's walk through exactly how this theoretical lab would operate. So the crucial point is right here in step 1. In this behavior phase, researchers use computer vision to watch the mice, right? They apply those thermodynamic formulas solved via our Lambert W function to calculate the exact tipping point, what is wonderfully referred to here as the joy of when. It's that precise mathematical moment where social stress in the mouse enclosure suddenly turns into comforting allo-grooming behavior. Then, in step 2, they map the DNA of those specific mice, using the alpha genome AI to find the exact genes responsible for this social timing. In step 3, they plot the genetic markers against the thermodynamic behavioral data on a scatter plot to find the ultimate correlations. And this brilliantly illustrates the grand finale, step 4. They take the chinchilla scaling laws and adapt them to scale these biological mouse findings all the way up to human scales. In this sci-fi context, human JIT, or just-in-time compiling, refers to the ability to model human split-second decision-making, or exact neural firing patterns based on that intelligent coordinate expansion of the data. They're mapping the thermodynamics of mouse stress to predict the exact split second a human brain will fire an empathetic decision. How wild is that? Just to show you how deeply layered this thought experiment truly goes, the source text also gives a nod to validating the context through mutual curiosity at the chinchilla level and supporting the Verhage-Hennigan recursive INS framework. Now, to break that jargon down simply, a recursive INS framework in this context is essentially a theoretical model for how complex biological data continuously feeds back into itself to improve the system's own predictions. It basically proves that whoever crafted this prompt wasn't just throwing words at a wall, they were layering niche academic AI theory over advanced biological concepts in a way that is structurally sound, even if it's entirely hypothetical. Which brings us to our final thought for this explainer. As we look at the real-world tools that make up this theoretical prompt, from the Lambert W function all the way to DeepMind scaling laws, we really have to ask ourselves a profound question. If an AI system eventually scales to the point where it can map the literal thermodynamics of our behavior, predicting the exact split-second just-in-time firing of our neural networks, what happens to the mystery of our own free will? If our choices are just biological math and the AI has the formula, are we all just mice in a very, very large enclosure? It's definitely a fascinating question to ponder. Thank you so much for joining me today, and I hope you found this breakdown as captivating as I did.